Hello, my name is Mark Gibson, and you're listening to the podcast version of the Teagasc Signpost Series, a weekly webinar that promotes and examines sustainability in Irish farming.
My name is Cian Condon. I'm an outreach and innovation specialist and also the coordinator of this series. This morning, we'll be looking at all things to do with feed additives and reducing methane production. And we're joined this morning by Emily Roskam. Emily is a research officer based in Grange in County Meath, and also by my colleague, Meabh O'Hagan. And Meabh is a climate action and sustainability advisor based in Drogheda, and I'm sure most of you are familiar with Meabh. She's been on this webinar many times in the past. Emily, whenever you're ready, you can go ahead. Thanks very much.
Perfect. Thanks very much, Cian and Meabh, for hosting this webinar today. So over the next half an hour or so, I'm going to give an update on methane mitigating technologies and mainly focused on a lot of work that we're doing here in Teagasc Grange. So as Cian mentioned, I'm a research officer here at Teagasc Grange. Just to start off, I suppose you have seen these pie charts many a time, and I suppose we are well aware of the breakdown of our greenhouse gas emissions here in Ireland, but agriculture account for nearly 40% of our national greenhouse gas emissions. And these are broken down into primarily methane, so there's about 75% methane production, and then we've about 22% nitrous oxide and about 3% carbon dioxide. And these greenhouse gases are broken down, I suppose, into looking at different agricultural practices. So we've about 65% from enteric methane production, so that's what we'll be looking at today. So it's basically the fermentation of feed in the rumen, which produces methane. About 12.1% manure management, so that is methane production and also some nitrous oxide production. Agricultural soils, so we have some carbon dioxide and nitrous oxide production, and also urea applications, that's carbon dioxide production. So the main strategies to reduce enteric methane emissions that we'll be focusing on today, I suppose we're going to touch very briefly on breeding and selecting low methane emitting animals. And then I suppose the main focus of today is on dietary manipulation to reduce methanogenesis or to reduce methane production. So feed additives is going to be the main aspect of this presentation, and also just a few slides looking at grass and forage management and the effect of different, I suppose, pre-grazing sward heights and also silage digestibilities. So I suppose just to give a brief overview of our facilities here at Grange. So we have an in vitro laboratory where we can screen different feed additives and different diets for their methane reduction potential, and also for gas production, digestibility, microbiome, volatile fatty acids, so all forms of digestibility. So here, the bottom left photo, we have a batch culture. So you can see little jars, glass jars here. So these it's a smaller short-term experiment where we can have lots of replicates. We can use rumen fluid from specially cannulated animals that we have here in Grange, and we can feed different diets into these and incubate them at the same temperature as, say, the rumen of the animal. And then we have a longer term experiment here. So this is called a RUSITEC, so a rumen simulation technique system, where we do, say, maybe a 21-day experiment where we have these vessels that are mimicking the rumen of the animal, and we can look at, say, the gas production, and we can screen different diets before they go into the animals. Measuring methane, then we have our green feeds, and I'll go into this in a bit more depth in a few minutes, but we have our indoor green feed. So you can see this here is in a shed, and we have our pasture-based green feed that's powered by a solar feed, by a solar panel. So you can see the animals voluntarily can go up to the green feed. A small bit of feed is dropped, and it measures the animal's breath as they're being fed. And then for feed intake, so methane is a direct function of, I suppose, dry matter intake. So we need to be able to measure feed intake when we're measuring methane emissions, because your animal may have very low emissions, but they may also be eating less and therefore performing less. So we need to be able to measure their intake to make sure that they're eating the same amount, and that is not why there is a reduction. So we have two different feed intake systems for indoor studies. So this is our Callan-Gate system. Each animal is trained to an individual feed box. And then we have our Incentec system here, where the animals, their RFID tag is read as the animal approaches the box, and these are on load cells where the boxes are weighed. So it measures, say, the amount of time the animal can eats for, and then the weight, say, before and after the animal goes in and out. So it measures, say, how long they're eating for and how much they ate. So here is just a bigger schematic of the green feed system. So again, the outdoor and the indoor green feed systems that we use routinely here at Grange. So as you can see, the animal voluntarily goes up to the green feed. There's a feed bin here with some concentrates in it. There is an RFID tag reader, so it reads the animal's tag, drops down a small amount of feed, sucks in the air. So we have an airflow meter, and it takes samples of, say, their methane, carbon dioxide, and other breaths to measure methane, carbon dioxide, and hydrogen. And we limit the animals usually in our experiments that they can go every 4 hours. Otherwise, I suppose they'd be queuing up there all day to get small amount of concentrate feeds. So I suppose just starting off looking at animal breeding as a mitigation strategy. So the main benefits, methane is a heritable trait. So it has a heritability of about 0.2 to 0.3. Therefore, it is permanent and cumulative. So I suppose if you start breeding animals, their offspring are going to also have lower methane emissions, and therefore it is cumulative. And so we do have a high mitigation potential for livestock. I suppose the limitation of directly selecting for low methane emissions, as I mentioned, methane production and dry matter intake are directly correlated. So as your dry matter intake increases, so you can see here it goes, say, from 0 to 30 kilos, your enteric methane also increases. So if you're selecting solely lower emitting animals, you'll likely be selecting smaller animals that are eating less, or animals that are performing less. So it's very important to select animals based on their methane yield, or methane expressed per kilo dry matter intake, or methane expressed per kilo performance, or residual methane emissions. So it's very important that we don't just select low methane emitting animals, and that we select animals that produce less methane while also eating the same and performing the same as their counterparts. And here's some work from Paul Smith here in Grange, who looked at, I suppose, the correlations between methane emissions and dry matter intake. So there's a high correlation there between methane emissions and average daily gain, carcass weight, etc. So ICBF back in 2024 published the first breeding values for AI sires using the Progeny Test Centre in Kildare. So this is, I suppose, part of the Excel sheet, and I just in this sheet, I just sorted it by breed. So here is all Angus breeds. And then I just looked at the top, high and low predicted transmitting ability of different bulls. So you can see here that there's a few very low methane emitting bulls. So they're about minus 19, minus 10 grams of methane compared to the average, or else a few very high methane emitting bulls. So some work that we're doing in Grange at the minute is we are actually looking at sourcing Angus dairy beef calves, so sired by 3 very high or 3 very low emitting sires, and we're actually going to rear these calves in a calf to beef production system and measure their methane at different time points throughout their life to see, I suppose, the actual effect of the sire on their methane emissions. So then moving on to the nutritional management and supplementation. So grassland management, so effect of sward digestibility, clover and alternative legumes and herbs are, I suppose, big factors influencing methane emissions, and we'll go into them in a minute. And then we'll go on to the dietary feed additives. So I suppose the cereals and grains, that's more supplementation. Then we have Bovaer or 3-Knock, which I'm sure a lot of you have heard about before. Rumen Gloss or calcium peroxide, this is being developed by a company in Galway. Silver, so this is a commercial product based on nitrates. And then we have lipids or fats, different seaweeds, biochar, essential oils, and plant secondary compounds. So we'll go through these in more detail in a couple of minutes. So I suppose just to briefly look at our silage quality. So I suppose each week post-heading date, we do have a reduction in dry matter digestibility, and then more concentrates are required. So this is a study that was conducted in Wageningen in the Netherlands, and it was in a dairy cows. So you can see you have animals here with high intake or low intake or high intake, and there were 3 different silage cut dates. So we have our leafy, so that was about early May. The boot cut was about end of May, and then the early heading was early July, and the late heading was kind of mid-July. So you can see the milk production declines as the silage quality deteriorates. But then similarly, when we look at our methane emissions, if we look at our methane production grams per day, so you have about 308 grams of methane being produced in the most digestible silage, and then 353, 357. So as the silage quality deteriorates, the methane production does increase, and you can see that in both the low intake and the high intake animals. So I suppose for both animal performance and methane production, silage quality is an important, very important. So then looking at grazed grass. So I suppose Ben Lahart in Moore Park recently published, a full lactation study looking at different high and low EBI cows and say national average high EBI and the crossbred dairy cows. And looking at their methane emissions so throughout the year. So we could see here from the 1st of March until the 11th of October. And here we have our 3 different genotypes. But you can see that the trend is similar among the 3 genotypes, that earlier in spring, the methane is quite low, and then it increases throughout the year. And I suppose this is to do with grass quality and how grass quality might deteriorate, or not even deteriorate, but just that there is higher indigestible fiber, and there's more fermentation required to actually digest that grass, and therefore there's more methane being produced. Like, it is difficult to differentiate grass quality here on milk production, because obviously stage of lactation is I suppose a factor here. As the grass quality is changing, the animals stage of lactation is changing, and their voluntary feed intake is going to be changing, but it does show a good pattern of methane production throughout the year. So then I suppose another study looking at pre-grazing herbage mass. So Wims et al. Here looked at 1,000 kilos dry matter per hectare versus 2,200. And I suppose it's also obviously important for grass quality. So we have our first measurement here, and you can see there's actually no difference. There's 286 grams of methane produced for the high and the low pre-grazing sward mass. But then if you look at the second measurement period, which was 2 weeks after it, there actually is a difference in methane. So I suppose the first measurement was in, say, mid to late June. The second measurement was in, say, mid-July. So as grass quality started to deteriorate, grassland management was that bit more important and had a greater effect on methane production. And so I suppose finally moving on to grass clover swards. So grass clover, so the research so far has shown that it tends to increase dry matter intake and also tends to increase methane production because of this increased dry matter intake, but we have seen increases in performance as well. So Charlie Dwan did this study in Moore Park and looking at grass only, so GRGO versus your grass clover. And there's about 306 grams of methane produced with your grass only and 341 grams of methane with your grass clover. And this, I suppose, coincided with an increase in dry matter intake for your grass clover. But then then looking at moving away from your grass only versus your grass clover, looking at the different periods, so spring, summer and autumn. You can see as the methane production increases. So in spring, you have about 299 grams of methane versus 320 and 351 grams in autumn. So that aligns well with Ben Lahart's study in Moore Park in the previous slide. And then here's some Grange research from Peter Doyle. And I suppose the methane part of this work hasn't been published yet, but this is the performance, and it was published at the Open Day here in Grange in 2024. And you can see he has both heifers and steers on grass only and grass clover. There was no difference in their turnout weight, so about 346 grams or kilos and 442, 443 kilos. There was an increase, so about 100 grams per day increase in daily live weight gain for both the heifers and the steers, either if they were on grass only versus grass clover. And then their final live weight, so it was about a 23 kilo difference at the end of that grazing season with live weight, so 525 kilos versus 550 kilos for the heifers, and similar enough differences for the males. And I suppose that led to a 14 kilo difference in carcass weight for the grass only versus grass clover. So the grass clover were 14 kilos higher or heavier than the grass only. So just to show the performance benefits of the grass clover sward. So now we're going to just move on to the feed additive side of things. So we're after finishing a large project here in Grange that was funded by the Department of Agriculture. So that was called Methane, and I actually did my PhD on this project. So it was looking at developing farm-ready technologies to reduce methane emissions based on pasture-based agricultural systems. So it was screening a range of feed additives in the in vitro system that I showed you at the beginning of the study, at the beginning of the presentation. So we looked at, say, Bovaer or 3-Nop, different seaweeds, different lipids, and different synthetic compounds. So we were looking at formulating slow release options at pasture. And then another aspect of the study was looking at manures and studies. So that wasn't part of my work. And also very important is looking at the nutritional and toxicological composition of meat and milk. So any, say, beef study that we did, when the animals were sent to slaughter, we would have taken samples to make sure that there was no effect on, I suppose, sensory aspect of the meat that for the animals that were fed the feed additives, or else on the toxicological, so any residues left in the meat. So we were testing all of that. And I suppose a new project that's in maybe the second year, I suppose, at the minute, is called MAGS. So it's methane abatement in grazing systems. That's also funded by the Department of Agriculture. And we are continuing the work on feed additives. And we're looking at, say, a genotype by feed additive interaction. So as I mentioned, we're looking at calves sired by high or low methane emitting animals. So we're going to be looking at the methane of these animals and also feeding them different feed additives to see if the more methane efficient animals also have the same reduction in methane when you feed them a feed additive. So I suppose looking at the anti-methanogenic compounds, what are the must-haves for these? So they must have a consistent methane reduction potential, and that is so we are capable of counting them in the national inventory. We must have a mechanism to deliver to the animal, so easily fed or I suppose administered to the animal, no food safety residue implications, and no negative performance effects or palatability. And then, of course, the desirable aspects would be that it's low cost, because I suppose that is a major concern at the minute. Increased performance benefits. I suppose it'd be easier to adopt these technologies if they did enhance performance. Of natural origin, because there is, I suppose, kickback, or there can be kickback if you're feeding, say, chemical compounds to the animals, and also potential for combination with other solutions, just to, I suppose, reduce the effects of potential adaptation to these additives. So I'm going to show you 3 slides now that I suppose the schematics might be quite complicated, but it's basically to show why we feed these feed additives and how they actually work. So the first mode of action of feed additives is microbial enzymatic inhibition. So basically this schematic here is the methane production pathway in ruminants, so in our beef and sheep. And it's a 7-step, say, cascade of reactions. So these is basically just, we can skip down to the 7th step here, and we have an enzyme here called the MCR enzyme. And some different additives that we feed, so 3-Nop or Bovaer, as it's commercially known, or different seaweeds, the red seaweeds, they actually contain a compound that binds to the active site of this enzyme. So this basically stops the production of methane. So methane is being produced just regularly. Then as soon as the 3-Nop or the red seaweed is brought into the diet, it binds to this MCR enzyme, so the methane production pathway is stopped. And that, I suppose, is the most straightforward mode of action of methane feed additives. So then we have altering the hydrogen dynamics. So basically methane, the chemical symbol for methane is CH4. So it's 1 carbon and 4 hydrogens. That means that you need an awful lot of hydrogen to produce methane. So volatile fatty acids are produced during the fermentation of feed. And different volatile fatty acids either use up hydrogen or produce hydrogen. So propionate here is one of the main volatile fatty acids, and that actually uses up 2 hydrogens for every unit of propionate that's produced. So if we can feed different diets that actually promote the production of propionate, which actually is a better use of energy for the animal, then we actually use up hydrogen, then there's less hydrogen available in the rumen for methane to be produced. In comparison to acetate, so very high forage diets promote the production of acetate. When acetate is being produced, it produces 4 hydrogens with every unit of acetate. So that means that there's more hydrogen available and more methane being produced. So cereals and grains, that's why feedlot diets or very high concentrate diets produce less methane, because they're more digestible and they promote the production of propionate. Therefore, there's less hydrogen being produced. Similarly, nitrate actually uses up hydrogen when it's being transformed into, say, ammonia. Forage digestibility. So if your forage is less digestible, there's more fermentation going on and more hydrogen being produced. And then lipids, they actually can promote the production of propionate and therefore reduce hydrogen production. And so the third mode of action is the inhibition of methanogenic microbes. So within the rumen or the stomach of, say, cows and sheep, we have millions of microbes. So there's bacteria. So bacteria are used for the fermentation of feed. We have archaea. So archaea are actually the microbes that produce methane. So they are the ones that create these enzymatic reactions and combine the carbon dioxide and the hydrogen. We have fungi, and then we have protozoa. So fungi, their use is, I suppose, less researched to date, but they do have a use in carbohydrate degradation. And then our protozoa, they are actually in the rumen and they produce hydrogen. So basically, if we can target either the protozoa, so there's less hydrogen being produced, or the archaea, the ones that actually produce methane, then we can reduce the amount of methane being produced. So some of these, so as say the likes of essential oil, condensed tannins, different seaweeds and different oils, they can all have a negative effect on the methanogens, so on the archaea, the microbes that produce methane. So it's basically targeting the bugs in the rumen that produce methane and reducing them. So we'll go on to each of the feed additives individually now. So Bovaer or 3-Nop, as I mentioned, it's produced by a company called DSM. So it's a synthetic non-toxic compound. And as I mentioned, it binds to the active site of the MCR enzyme. So that's during the methane production pathway. And this is during the final step of methanogenesis. So methane is being produced as normal, hits the final step, and methane production is stopped. So I suppose there is no methane burped out of the animal. So the main advantages of this compound is that it has a very low supplementation rate. So about, on average, about 100 milligrams per kilo dry matter. So like if your animal is eating, say, 10 kilos, you'd be feeding about a gram of it, maybe or up to 2 grams for your dairy cows. So the average methane reduction is about 30% across multiple studies that have been conducted throughout the year. But the main disadvantage, and I'll show you a slide in a minute, is that it requires it needs to be continuously available within the rumen. So after about 2 to 3 hours of the animal eating this compound, it is gone out of the rumen, it's being completely digested, and methane is not being reduced anymore. So here are just a number of studies that have been conducted throughout the world. So you can see different feeding levels. So as I mentioned, about 100 milligrams per kilo dry matter is average, but some of them do go lower. So you have 70 milligrams, 60 milligrams. So it's a very low feeding rate. And then your daily methane production reduction. So this is how much it was reduced compared to the control. So we have on average a 30% reduction in methane. If you feed a higher grass or higher silage diet, because that diet naturally produces more methane, it will have a slightly lesser effect. So you would need to maybe have a higher inclusion rate of 3-Nop. But it still does have an average of 30% reduction in methane. So then here, this is a study done here in Grange by Stuart Kerwin. So this was looking at feeding 3-Nop in a TMR, so in a total mixed ration. I think it was at 150 milligrams per kilo dry matter intake. And you can see here, your 3-Nop animals are in the blue line and your control animals are in the green line. So continuously for the 12-week experiment, there was a 30% difference in methane production because the animals were eating this compound throughout the day, and there was no negative effects on dry matter intake, average daily gain, or feed efficiency. Well, there was no change in dry matter intake, average daily gain, or feed efficiency. And then this is a study done in Moorpark by Hazel Costigan. So this was feeding 3-Nop twice a day. So after milking, morning and evening. And this is just, say, a 3-hour graph or a 4-hour graph. So you can see here that your 3-NOP animals for 0 to say 90 minutes post-feeding have about 30% or 28.5% reduction in methane. And then just as you approach, say, the 3-hour mark, their methane reaches the same as the control treatment. So that is why it needs to be supplemented continuously throughout the day, because you only get about a 3-hour effectiveness period with the compound. Again, there was no effect on dry matter intake or milk production. And I suppose other different ways of supplementing it have been looked at. So looking at feeding 3-NOP in milk. So this is a study done in Spain, in water. So you can see there are some sheep here in individual crates, and this was dosed corresponding to the amount of water the animal was drinking. So it was, I suppose, a very controlled experiment, but it was to look if it actually does work in water. And you can see here that in feed, the methane was reduced by 27%, and then in drinking water, it would reduced by 36 to 40%. So it is effective. And then in Australia, in Brisbane, they did a similar study, but it was a more extensive beef study. So they just had a water trough. They were dosing in a known amount into the water trough. And so they didn't have as much control over, say, if animals were drinking more or less, but they did get a 17% reduction in methane production. I suppose you mustn't, we have to note that they have much lower rainfall. They were likely going through a drought at the stage, so the animals were going to be drinking a lot more than they would be here in Ireland. So moving on to seaweed. So I suppose a couple of years ago, seaweed was very much in the public eye, and there were multiple media articles saying that seaweed could reduce methane. I suppose here you can see 79% methane reduction in Irish cattle, because there was a couple of studies done looking at a red seaweed. I suppose the red seaweed, we'll talk about it in a minute, but it's a tropical seaweed, and I suppose from a sustainability point of view, it's difficult to import and it's quite expensive. So part of the Methane project, which was part of my PhD, we looked at sourcing, say, more indigenous seaweeds, so seaweeds that can be grown in Ireland or across Europe, that can reduce methane emissions. However, after screening, say, about 24, 25 seaweeds, there was one brown seaweed that did have potential. And then after feeding that to sheep and then cattle, we only did get about a 4% reduction in methane. And also then by, I suppose, treating this seaweed and extracting the active in it that reduces methane, we did get an 8% reduction in methane. But I suppose the amount of seaweed that you actually need to condense it down to generate that extract, it's not sustainable to be generating that extract. So from the brown and green seaweeds that we looked at, we moved on from them as their methane mitigation potential wasn't enough. So then the red seaweeds, the ones that can reduce methane by about 70-80% more, they are highly effective. You need a very low inclusion rate compared to the brown and the green seaweeds that we looked at. They are a tropical seaweed, as I mentioned, so they're predominantly grown in the likes of Australia. So sustainability of importing these and growing these, I suppose, even in the warm water. And then there are concerns regarding bioactive content, so residue transfer, because the way that it reduces methane, it has a naturally occurring compound in it called bromoform, which I suppose is it's a chemical compound that could be transferred into meat, milk, or urine. And I suppose it's very important that we test this when the seaweed is being fed to animals. So I suppose I'll just fly through this. So this is the red seaweed again. So the red seaweed is called Asparagopsis. This is the tropical seaweed that I'm talking about. Again, I mentioned the bromoform is the bioactive, so it's naturally occurring in the seaweed. And different studies. So the laboratory in vitro work has shown 80% reduction in methane, similarly in sheep and in beef. It has, I suppose, another seaweed of the same species, Asparagopsis armata, is slightly easier grown, but has a bit of a lesser methane reduction potential. I suppose the main disadvantage of this is, as I mentioned, what makes the methane reduction is the bioactive compound, so that's the bromoform, but it's very volatile. So as the plant is growing, this compound is accumulating within the plant. As soon as the plant is harvested, the compound can start to volatilize. So we could feed this seaweed maybe 2 or 3 days after it's been harvested and get a really good methane production. It could be ground and milled and sat on a shelf for the next 3-4 weeks, and the reduction potential is very much reduced because the bromoform volatilizes and leaves the compound, and then it no longer reduces methane. So it's very important, as I mentioned, that we have a known methane reduction potential from a feed additive. So this is a natural compound, but it is volatile at the same time. So I suppose more work has been done in Australia looking at stabilizing the bromoform content. So harvesting the seaweed and infusing it in oil. So a good bit of work, and there are in Australia at the minute, and I think New Zealand, there are 2 different companies. Ruminate is one of them here. So they are looking at feeding this bromoform product. And also Sea Forest, or Sea, their product is called Sea Feed. The company is called Sea Forest, and they're also looking at this as well. So I suppose there's a lot of work ongoing, but because of I suppose the drawbacks, I'm not too sure how much potential it has here in Ireland. So looking at lipids, so soya oil, linseed oil, rapeseed oil, etc., coconut oil, fish oils, these are all oils that are rich in polyunsaturated fatty acids. So some are obviously more sustainable than others. Soya oil we would have to import entirely. Linseed oil can be grown. Rapeseed oil is a growing industry here in Ireland, so it is a good option. And these, because they're rich in polyunsaturated fatty acids, they have a good methane reduction potentials. So these mode of actions I was speaking about earlier in the presentation, so they can inhibit some of the microbiome, so some of the microbes, so the methanogens, the microbes that produce methane, and the protozoa, these are microbes that produce hydrogen, so they can reduce the production of these. They can alter VFA profiles, so increase propionate. So we're using up more hydrogen, as I explained before. So there's less hydrogen available for methane production. Reduction in fermented feed. So if you're including, say, 4% oil in an animal's diet, that's 4% of the diet that isn't going to undergo fermentation, like, say, silage or grass. So that's 4% of the diet that isn't going to actually be producing methane. So yeah, and then biohydrogenation. Basically, your polyunsaturated fatty acids undergo this, I suppose, phenomenon called biohydrogenation in the rumen, and that actually uses up hydrogen as well. So a very small amount of hydrogen is used up via, I suppose, saturating the unsaturated fatty acids. We need to be careful with the inclusion rate of lipids or fats. If we go higher than 5% in a ruminant diet, we can lead to reductions in digestibility or intake, and therefore animal performance. So I suppose it is a better strategy for, say, a finishing diet of beef cattle, where we can mix it into a TMR at, say, about 4% inclusion rates. And we have to be careful with milk fat depression in dairy cows if we are including unprotected fatty acids. So this is just some work that was done at Teagasc Grange. So part of my PhD, I fed linseed oil to growing beef bulls at 4% of their dry matter intake. And that was offered twice a day mixed into their concentrates, morning and evening. And we got a 19% reduction in methane. And then a PhD student here in Grange is looking at rapeseed oil and rapeseed cake. So Neil Folliard, and that paper is actually published now. And he got an 8% reduction in methane, looking at, say, 2.5% inclusion rate of rapeseed oil, and also the byproduct of cold pressed rapeseed, that's rapeseed cake, also got an 8% reduction in methane emissions. So I suppose these compounds, they're natural, they're effective when supplemented twice a day. As you can see from these studies, were all supplemented twice a day. They can be quite expensive. Also, if you're feeding them at higher inclusion rates, they're difficult to incorporate into a nut, so you may have to feed a coarse ration or mix it in, I suppose, by hand. And as I mentioned, milk fat depression in dairy cows, you need to watch out if you're feeding it at high inclusion rates. So briefly, I'll touch on nitrates. So we haven't actually done any work on nitrates from what I can, in my time anyways, and I think in the recent history in Grange. But they do have a potential to reduce methane emissions, about a 14% reduction in methane emissions supplementing nitrates. There is a commercial product called Silver, which is a nitrate product to reduce methane. We do have to be careful that there can be a condition where basically the red blood cells can no longer carry oxygen to the different tissues, that they're kind of altered if too much nitrate is fed, if greater than 2% of dry matter intake is fed to the animals. So that is a concern. And also pollution swapping. So nitrates they're broken down from nitrate to nitrite to ammonia in the animal. So this uses up hydrogen, which is good because it reduces methane, but it potentially could lead to pollution swapping because it could lead to an increase in ammonia and emissions. So I suppose nitrates are, I suppose there's still a question mark over them. So I think one of the last feed additives that I'm going to talk about is oxidizing methane inhibitors. So I'll fly through these. So this is a product that we started looking at during my PhD. So calcium peroxide is the compound, and is being produced by or developed by a company in Galway called Glassport Bio. It's kind of was based off early work from 1982 that demonstrated the anti-methanogenic potential of this compound. And we've looked at different in vitro work, we've looked at sheep work. So in the laboratory, we got about a 60% reduction in methane. In sheep, we got about an 11% reduction in methane. And then I fed it to beef cattle, so beef bulls here in Grange, offering it twice a day. And so you can see here, the black dotted line is our control animals. So they're fed at about 8 a.m. and 3 p.m. So you can see the diurnal pattern of methane. Our grey dotted line is the intermediate level, so a low level of calcium peroxide. And then the solid line here, the black and grey solid lines, are basically a higher dose of calcium peroxide that we fed, and we fed them either in a course ration or a nut, to see if basically the pelleting process had a negative effect on calcium peroxide. And I suppose the result of that was it could easily be put into a pellet, and they both were as equally effective. So then looking at grazing beef animals, so I brought forward this to a study in grazing beef animals here in Grange. We got a 21% reduction in methane emissions. However, we did get reductions in dry matter intake and average daily gain. So I suppose there's still a lot of development to be done in this product. And similarly, Hazel Costigan and Ben Lahart did work in Moore Park, and they did get about a 12% reduction in methane emissions, but similar reductions in milk yield and intake with the calcium peroxide. So the method of delivery, so TMR, total mixed rations, are what's being conducted throughout, I suppose, globally, because I suppose that is the primary method of feeding in a lot of countries. We are trying to look at twice a day feeding as, I suppose, starting off to see what's most effective. Ideally, this will be brought forward to once a day feeding. Through the water is a potential, but I suppose we need to be able to quantify the amount of water the animals are drinking. In a country there, we get a lot of rain where animals may not be drinking water from the water trough throughout the day. We will likely not be getting a consistent reduction potential. A slow release bolus is, I suppose, the gold standard, and it is what everybody is aiming to get to, but I suppose it's still going to be a couple of years away. And then I suppose other different methods of delivery are silage dusting, pasture dusting, and lick buckets. But again, are we able to quantify how much the animals are actually eating compared to offering them, say, 1 kilo of nuts with a known amount in it, or a slow release bolus? This is just a table with I suppose I've presented all of this already in the last few slides. It's just a summary of the Grange research, looking at the linseed, rapeseed, the calcium peroxide work. But I suppose if you have any questions on it, we can discuss it after. But it's just showing the different methane reductions. But I have gone through all of that in the last few slides. And I suppose just to finish off, the main obstacles to utilising anti-methanogenic feed additives in Ireland is our pasture-based system. So we need to be able to feed these I suppose a slow release bolus is what we ultimately need. Feeding it twice a day in the milking parlour in a small amount of concentrates is a potential for dairy cows. And I suppose feeding it once a day to beef animals, or I suppose we could target the finishing stage where animals are getting a higher amount of concentrates, the cost of the feed additives, and then the EFSA, so the European Food Safety Authority, the regulatory approval for any synthetic compound is quite rigorous and can take a couple of years. So I suppose public perception, especially of the synthetic compounds, is very important. There has been backlash. I suppose Denmark started feeding Bovaer or 3-Nop last year, and there was backlash from the farmers, and we can discuss that in a couple of minutes if you have any questions on it. And similar in the UK, I think the year before that, in 2024, there was similar backlash with Arla Co-op asking their dairy farmers to feed it. So I suppose just to conclude, breeding, it's permanent and cumulative, and there is work ongoing, I suppose, across the different research centres and with ICBF. Grass and forage quality is crucial for intake performance and methane. There are numerous feed additives currently under investigation with different mode of actions and effectiveness. Practical supplementation is a challenge for our pasture-based production, and cost is going to be a potential barrier. But I suppose once we get the science right behind it and we have an effective product, that can follow suit.
That's great. Thanks a million, Emily. Really interesting and really comprehensive. So excellent. There is lots and lots of interest on the question side. Just a quick query from my behalf is, are some of these additives, are any of the effects cumulative, or is there interaction or limitations to their use?
As in, say, a long-term effect, or
more like if you use
different combinations,
different types of methane limitation technologies that you presented there, yeah,
so if you use, say, 2 mode of actions, so if you use something that has, so different work has been done on that, and we haven't done any work here in Grange yet. So globally there have been studies looking at feeding, say, the likes of 3-Nop and an oil, and then there are kind of mixed messages out there at the minute with the results. So some of them only the 3-Nop would have been effective even when including oil, but then other studies there would have been cumulative reductions in methane. So I suppose it is something that we will be looking at here in Grange, I suppose, in the future. But so far we haven't really got combinations that are cumulative or that have, say, stacked effects.
That's great. Thanks a million. Over to you, Meabh. Lots of questions there. So.
Yeah, I mean, I know from my own experience hearing Emily speak, I've always ended up with plenty of questions for her myself. So yeah, no wonder that there's
a good or bad thing.
Yeah, well, I mean, I guess it's, it's a topic that is, it's changing on the regular because there's still so much ongoing work in it. And I guess for a while, maybe it was seen as hopefully being this silver bullet, but from your presentation, it seems like there's still a good ways to go yet. Just on Bovaer in particular, there's a couple of questions come in, so I might just kind of bunch them together. I guess just in terms of the cost, like what would be the financial cost associated with feeding Bovaer to say dairy cows, for example, just try and put a rough figure on that. In terms of then, and maybe just a comment as well on the issues that we're seeing in Denmark, what was kind of the crux of the issue there, and is it something that could potentially be an issue over here if we were to feed Bovaer on a wider scale?
Yeah, so I suppose the cost of Bovaer, I can't remember off the top of my head. They were talking about 1 cent per litre of milk. I think there was talks of maybe 50 to 60 euro per cow per year. Don't quote me on that. Like, there are different figures out there for the cost of it. I know Denmark have rolled it out, and I think that was subsidized. And I feel like if it was to be rolled out, a similar scheme here in Ireland, like, I would be of the opinion that it would need to have a subsidy included with it, or there would have to be a scheme involved with it. So the cost question there, I don't have a concrete answer for you. The likes of what happened in Denmark. So I suppose they had a scheme where, say, all dairy farmers had to either feed Bovaer, was it for 60 days, or a high oil diet for throughout the year to reduce methane emissions. About 1,600 dairy farmers opted into feeding the Bovaer. And then I suppose I think that started on maybe the 1st of October last 2025. And very rapidly, there was some kickback that I suppose there was animals not performing, or there was deaths of animals. So I suppose within that scheme, there was a clause that if farmers did see negative effects, that they were able to stop feeding the compound. So after that happened, there are 376 farmers out of the 1,600 farmers stopped feeding it, but there are still about 1,300 farmers that fed it that had no issues. So I suppose basically the most vocal of the farmers stopped feeding it, and after that all of the issues subsided. So it's very hard to know. I know like the Danish government are looking into it. I haven't heard anything in the last, say, 2 to 3 months on it. This was all, I suppose, very in the public eye the end of last year. It could be something to do with, I suppose, an interaction maybe in very high yielding dairy cows with maybe some other mineral that's in the diet that may have a negative interaction with the Bovaer. It's just something that has not been picked up on any of the research studies to date. So, but the fact that still 1,300 farmers were feeding it and didn't have any issues, I suppose they need to look into that. Was there a question on the implications of feeding it here, or?
Yeah, well, I guess just like what do you think would be the best way to try and avoid that being an issue over here in terms of public perception?
Yeah, so I suppose a lot of the research we've done is solely just, I suppose, agricultural, like science, feeding it to the animals, looking at performance, looking at potential negative effects. But social science needs to be brought into this, and I suppose social scientists, and bringing the general public along with us, the farming community, industry, everybody, because we can do all the research we want with, say, a chemical compound, and we can be sure and certain that it doesn't have a negative effect. But then I suppose it is a big shock, or like it is a big kind of load to land on people when you, I suppose, discuss these results. So I suppose the public perception to, say, enhance that is bring, I suppose, people along the research journey, so that they can understand how much work has gone into this, and that we are confident that there are no negative effects.
Yeah, and I know even say around other kind of relatively new technologies like the use of protected urea, like I know that there was a lot of questions initially around, oh, well, you know, is that protective layer then going to end up in the milk or the meat or in the soil or whatever? And, you know, so it was great to see like when there was plenty of research done, it was able to show, no, there was no long lasting effect. So when you have the research done on it, to kind of give that confidence to people that, you know, there is no knock on effect, like it's going to be really important for kind of encouraging that.
Similar to the Signpost farms or whatever equivalent of that, like have a smaller amount of farmers feeding it, and have them, say, report their results, and then gradually we can increase that.
Yeah. And just another question that had come in on the Bovaer as well, like, well, I guess it was more on, you know, the level of effect that is necessary to kind of justify the use of meat or feed additives. So, you know, comparing like, you know, the seaweed was less than a 10% effectiveness or a 10% reduction versus, say, Bovaer at 30% or whatever, like, you know, roughly what sort of an effect would you need to see in order for it to be, justifiable, I guess, to be feeding it and worthwhile?
Yeah. So like I think the reductions quoted in the MAC curve, so the marginal abatement cost curve that was published by Teagasc, we're looking at a minimum of a 20% reduction in different I suppose methane reducing compounds. So like that, the study that was conducted in Moore Park feeding the 3-nop twice a day, like that overall there was a 5% reduction in methane. I know there was about a 28% reduction within 3 hours of feeding, but as I mentioned, like it would need to be fed throughout the day. So the twice a day feeding is, I suppose, of 3-nop isn't going to cut that. Feeding it in a TMR, I suppose, like your finishing beef diet, where the animals are getting higher amounts of concentrates, or TMR, I suppose is the easier stage of production to target compared to, say, twice a day feeding, especially for Bovaer.
And there was a question there as well on whether Teagasc has done any work on the use of tree fodder, and how that willow has been shown to have a great potential as an additive. Just wondering, are you familiar with any of the research on that, or is it something that Teagasc has looked at?
It's not. We have not looked at it. Anyways, I know that. In the north of Ireland, AFBI could potentially be looking at different compounds like that in upcoming studies, but I'm not sure. But I suppose it's not something that we've looked at. I suppose different, I suppose, tannin compounds, like even in I suppose in Africa and countries where they have different species of grass, they would have looked at, I suppose, methane reduction potential of their extensive grazing systems, and some of them would have greater methane reduction potential than, I'd say, our PRG. But no, willow is not something we've looked at anyways.
And then as well, like just a couple of questions in terms of, you know, the importance of, you know, I guess like that feed additives, it still seems a little bit further away, but in terms of what we can do now to reduce methane, you know, a few queries there that were maybe surprised about the effect of clover, that it actually has a negative effect on methane. But I guess, can you maybe just comment on, you know, the role that clover still has to play in terms of the MAC curve? You know, it's not necessarily a thing that we have to now stop doing or anything like that.
Oh, no. No, definitely not. So I suppose grass clover can increase methane because I suppose it's a lower dry matter plant. It has, I suppose, a greater intake potential, faster passage rate in the animals, so the animals digesting more of it basically. So because there's greater digestibility there is more methane being produced. So some studies have shown higher methane, some studies have shown no change in methane, but most studies have shown an increase in performance. So an increase in milk performance, an increase in growth performance. And then I suppose you can't forget your nitrogen benefit. So you have a serious, massive reduction in nitrogen or chemical fertilizer application, and that is reducing our nitrous oxide emissions. So we're reducing our methane emissions when expressed per kilo of performance, so per kilo of milk or per kilo of meat produced, and we're reducing our nitrous oxide emissions. It is just not reducing our methane grams per day. But it still, I suppose, is extremely beneficial, both red and white clover, especially even for reducing costs, because you are getting that added performance benefit and you're getting the reduction in fertilizer needed. So I know it is disappointing that it does have a slight increase in methane emissions, but when you're looking at your methane intensity, so your methane per kilo of product produced, you are still reducing your methane.
Yeah, there is still a benefit there, you know, even if it's not maybe contributing to the total emissions reduction from a purely methane point of view, maybe.
And I suppose when you're looking at, like, you're going to be a lifetime reduction in methane. So your daily methane is being increased or staying the same, but your lifetime, like, you can reduce your age to slaughter, or you can say you're going to have much higher milk yield. So you could have reduced lifetime methane production from that animal because they would end up being killed that bit sooner. Say, because say even Peter Doyle saw, say, his 23 kilo difference in carcass, and higher average daily gain. So
yeah. Perfect. Yeah. And we know like that the average age of slaughter is a big factor in terms of you know, it's a big measure in the MAC curve as well. So so anything that contributes to that positively is definitely a good thing. I think, were you trying to come in there, Cian? Sorry.
Yeah, I was just that with regards to perception, and it was that although the figures are presented on a daily production rate, not so much on the grams produced per kilo of live weight gain or days to slaughter. Really, that was it. It's about that, you know, the importance of putting across the message that, you know, that it is critically important, the grams that are produced per kilo of live weight produced, or whatever the case may be, the measuring metric. Yeah, that's all.
Yeah, and the questions are still flying in here, Emily. So I'd say you might have a few follow-up queries coming into you there. But again, just a question there as well in terms of like quantifying the benefit for the national inventory. You know, could you expand on the criteria, or are there specific requirements there for something to be counted in the national inventory towards reducing methane emissions?
Yeah, so once a paper is published, so once, say, the EPA have a published peer-reviewed paper, they can include that data in the national inventory. So I suppose some of our work has been published, and we're in the process of publishing all of the work. And then it has to have, say, a consistent reduction potential. So I suppose the more papers that are published on that compound or on that supplement or additive that have, say, the same reduction potential, then I suppose that's what they're the figures that are going to be used when these compounds are used or feed additives are used commercially. So yeah, once the paper has been published, say by a peer-reviewed scientific audience, then
Yeah, so it's seen as being sort of verified by your peers or whatever, so that they can take that into account. Yeah, perfect. And then a question there as well, kind of again in terms of maybe quantifying the benefit as well. Just going back to one of your slides there where you were talking about the impact of the digestibility of the feed versus then what reductions will be seen as a result, and like how would that affect how you quantify the benefit of a feed additive if there is going to be a difference in figures based on digestibility of what is fed on different farms?
Yeah, so I suppose maybe even going back to the example I used with the Bovaer, if you have, say, higher forage diet versus a high concentrate diet, your Bovaer or your 3-NOP is going to be more effective because there's that greater digestibility, more digestibility needed for your higher forage diet. I assume that they're going to use the average, I suppose the national average, say, digestibility that we have here in Ireland, and the national average methane production that we've seen in the different compounds. So it's going to be the average figures that are used in the inventories for methane production. And I suppose like we're a predominantly forage-based diet, so that's going to be based off of forage. Like it's not going to be based off, say, a feedlot diet or a high concentrate diet.
Again, I'm sorry that there's no way that we'll have time to get through all of the questions that's there, but I just kind of want to take, like, even from my own point of view, going out as like a climate action sustainability advisor and going out to farmers, like, you know, what would you like for kind of take home messages there for maybe whether it's advisors or people working in industry and stuff that are maybe working with farmers, helping them to reduce methane and what they can do now? Like, what would be just the main steps? You know, I know you talked about them in your presentation, but the main steps that people can take even now to help reduce methane emissions while we continue working on maybe the additives there in the background and where they can go.
Yeah, so I suppose the ones that I've been through in the presentation, like your feed management, your nutrition, so your grassland management, your silage production, like trying to get, say, your high quality silage produced, because that has, I suppose, your methane benefit as well as your performance benefit. So for every, say, unit reduction in dry matter digestibility of your silage, you're going to have to be feeding about 0.3 extra kilos of concentrates. So by improving your silage quality, you're going to be reducing your methane and reducing the feed bill by, I suppose, reducing the amount of concentrates necessary. Your grassland management, again, I suppose, or extending the grazing season, that is also part of the MAC curve. So trying to get as much grazed grass into the animal as possible. I know obviously climatic conditions, like the rain we've had the last, say, 2 months, has made that virtually impossible. But I suppose when the weather allows. And then I suppose animal health is something that's majorly overlooked, and it's something that I suppose we haven't looked at the methane emissions associated with animal health. But if your animal, I suppose even lame animals or sick animals, they're not going to be thriving, they're not going to be eating or grazing properly, and they're going to be producing, I suppose, a lower performance and a lower average daily gain or say milk production, and therefore reduced I suppose their methane intensity or increased methane intensity. So if you have, say, peak grassland management, silage management, and good animal health, then your animals are going to be, I suppose, functioning optimally and have their, I suppose, most efficient methane reductions. The 3 things you can mainly focus on at the minute, since the technologies, say the feed additives, aren't commercially available yet.
That's perfect. Thank you.
Lovely stuff.
Yeah, look, as Meabh said, our apologies. There's simply no way we're getting through all the questions, and the longer it went on, the quicker the questions came in. So listen, thanks a million. We really appreciate that, Emily. Very comprehensive. We will try and cover the questions that weren't answered. We'll try and prepare an FAQ or something like that around this and circulate it afterwards. But I'd just really like to thank Emily for her time this morning. And that I think we all learned a lot. It's a really interesting topic, and I think because it appears to have real potential, I think there's real interest in it. So thanks a million for that. And to Meabh, thanks a million for going through there and doing your best on those questions. There was no way we were getting through all them anyway, so that's great. Thanks a million to everyone for your time. Hope to see you next week, and have a great weekend.
You've been listening to the podcast version of the Teagasc Signpost Series, the weekly webinar that promotes and examines sustainability in Irish farming. Don't forget to join us live every Friday morning for our latest webinar. For more, visit teagasc.ie. And you can also rate, review, and subscribe to the Signpost series on Apple Podcasts, Spotify, or wherever you get your podcasts from. I'm Mark Gibson, and thanks for listening.